Welcome to Microbial Concepts. So this is another video related to pharma interview questions. That means if you are a microbiologist or biotechnologist and if you are going for a job interview in pharmaceutical industry, then from that perspective I have discussed some questions. Okay, so in this video we are just going to focus on API, that means active pharmaceutical ingredient. Already a video is uploaded where I have discussed questions related to fermentation, related to sterile water injectables and clean room. Okay, now in case of clean room, two separate videos in detail are uploaded. They are present on my channel, so you can go check it out. So let's start with API related pharma interview questions. So there are different types of products that a pharma industry produces. So one is API, that means a single active pharmaceutical ingredient, okay, a compound which actually gives therapeutic effects. Then sterile water injectables or sterile injectables or the complete drug is produced. Okay, so these are random questions which are based on microbiology. Okay, so this is to give you an idea what kind of questions you may get. Okay, so already as I said, a video is uploaded on fermentation, sterile water injectables and clean room. Now we are just going to focus on APIs. So what are active pharmaceutical ingredients? So API or active pharmaceutical ingredients, they are biologically active components in a pharmaceutical drug, okay, which actually provides therapeutic effect, right? So a very basic question. So if you are a fresher, then you will not have an idea about active pharmaceutical ingredients. So don't worry. So this is for experienced candidate. At least 1 year experienced candidate will get this type of question because you have entered a pharmaceutical industry. You are working there. So you must have heard about this term and you should know what is active pharmaceutical ingredient. Now, how are APIs produced? So this depends on the industry where you are applying. Okay, so not necessary that if the a particular unit is not working on API production but it is using a active pharmaceutical ingredient in drug manufacturing, then you may not get this question. This is not compulsory, but you should know that APIs, they can be produced by chemical synthesis, fermentation or extraction from natural sources. Now it depends on what kind of API is getting produced. So for example, if a particular antibiotic is getting produced, then it can be produced by using a fermentation process from a microorganism, or some hormones or steroids, they are produced by chemical synthesis. Okay, so that's the difference. It depends on what kind of product the active pharmaceutical ingredient is to be produced. Now what is the role of microbiology in API production? Okay, so this is very important, and as you are applying for this particular role, you should know that. So the role of a microbiologist or role of microbiology in API production is as follows. Microbiologists, they play a key role in production of API, particularly those which are produced by fermentation technology. Why? Because microorganisms are used to produce such kind of products like many antibiotics or enzymes and other therapeutic compounds. Okay, so where there are microorganisms, a microbiologist comes into the picture. Now they are involved in the selection and manipulation of these microorganisms to optimize the production of the desired compound, and also they are required in QC for quality testing. Okay, so this is the main role of a microbiologist or main role of our subject, that is microbiology. So how do you ensure the purity and quality of APIs? So purity and quality of API can be ensured through combination of testing and monitoring. That means a microbiologist is involved in testing for microbial contamination, presence of endotoxins, as well as other quality parameters such as purity, potency, stability, etc. So you have to do testing of your product, and you need a controlled monitoring throughout the process, and you need to maintain the consistency. Okay, so that's how you can maintain the quality and purity of your product. So what are some challenges which are associated with producing API? That means what are the challenges which one can or one has to deal with while production of API. So the very most important challenge is strict control over manufacturing process. Why? Because you need or there is need of consistent purity and quality, as we are going to use this API, that is active pharmaceutical ingredients, further into a particular drug, and that is to be administered further to a patient. So we cannot tolerate any kind of microbial contamination, because it can anyhow it will lead to some kind of problem in the patient. Okay, we don't know. So we need a strict control over manufacturing processes. Now, the potential of microbial contamination or other quality issues, we need a control over them, and the need to develop and optimize new production processes for novel compounds. As we know, microbes, they keep on changing, they keep on getting adapted to the newer environments, and they are getting resistance to different antibiotics. So just to give you an example here, why it is written that optimize new production process for novel compounds, that is also a challenge. Why? Because microbes, they are getting adapted and they are getting antibiotic resistant, right? So there is a need for screening and production of novel compounds. Okay, so for screening, lot of time is required, and after that you need to optimize that particular compound to get it produced or synthesized on large scale. So again, that is again a challenge. Okay, so after that, next question is, how do you stay up to date with new developments in API production? As you know, microbes, they keep on emerging, or not just microbes, there are other illnesses, diseases on which we need a drug to be produced. So new drugs, new API, that means active pharmaceutical ingredients, they are coming up. So how to stay up to date with these new developments? So staying up to date requires ongoing education, training, as well as keeping up with the scientific literature and attending conferences and other professional development opportunities. That means simply you need to be alert, you need to be uh, going through or reading a lot of scientific research, okay, and attending conferences, so that you can stay up to date with new developments. Then now purely moving towards microbiology related stuff, okay, which is done in quality control. So very important process is, or important test actually, is microbial limit test. It is also in short it is written as MLT, which stands for microbial limit test. So what is microbial limit test? So it is actually a test which is performed to determine total number of viable microorganisms present in your sample. Okay, that can be of a raw material, that means your sample can be of raw material, it can be intermediate product, that means it has still in or it is still in process. Okay, and finished product or any environmental surfaces that are to be monitored. Now microbial limit test, from this particular name itself you can uh, say that what is the limit for having microbial contamination in a sample. There is also acceptance criteria for that, and that is what we are testing here. So total number of viable microorganisms. Okay, we are just focusing on viable, that means one which are viable, which are able to grow. Okay, so this test is commonly used in pharmaceuticals, food, cosmetic industries to ensure that the product is safe for human use and that it meets required microbial or microbiological specifications. Okay, so what microorganisms are typically targeted in this MLT? So MLT is used to detect the presence of microorganisms. We have seen that we do this test to know what number of total viable microorganisms are present in the sample. So as we are saying microorganisms, that means bacteria and fungi. okay, in fungi, molds and yeast also. So we have to detect for their presence in the sample. The microorganisms targeted in this test, they may vary depending on the type of sample and specific requirements of the regulatory agency over that industry. Okay, so some of the common microorganisms that may be targeted are as follows. First is, this is compulsory, you have to note it down: total aerobic microbial count and total yeast and mold count. Okay, so you can make out difference between yeast and mold count and aerobic microbial count. There are different tests for that. Then if you get a question just on what pathogens are typically tested or targeted in MLT, then here is the list. First is E. coli, that is Escherichia coli, then Pseudomonas aeruginosa, then Staphylococcus aureus, Salmonella species, Bacillus species, Clostridium species, then Candida albicans and Aspergillus niger. Now it depends on the industry, or it also depends on the type of sample. Okay, you may find that one or two of these pathogens, they are not tested. Okay, for example, Clostridium species, um, it requires anaerobic jar and etc for its growth, so sometimes it is not tested. Okay, so it depends on the industry and the sample. So how do you interpret the results of microbial limit test? Now, interpretation of your results: you have performed your test. Okay, you have inoculated your samples in particular media, specific media, you have incubated, and now you find growth on your plate. So how to interpret the results? Now the interpretation, it depends on the type of product that is being tested and the specific requirements of the regulatory industry or the industrial standards. Okay, so it depends on this. So it may happen that uh, if you are an experienced candidate, then I cannot give a specific example here because it depends. The one who is watching this video, you may find difference. Okay, so I'm just giving you a brief idea here that it depends on the product that is being tested, specific requirements, by the specific requirements of regulatory agency and the industrial standard. Sorry. In general, the results are interpreted by comparing the number of colonies. Okay, so number of colonies, you have to count them, right? And the one which grows on your culture media, that means those are the viable microbes. Okay, and then you have to compare it with the acceptable limits for the particular product or environment. Okay, so the acceptable limits are specified, so we refer to the pharmacopoeia. Okay, US pharmacopoeia, European pharmacopoeia, and depending on that, we prepare SOP, and in that SOP acceptable limit is given. Okay, so we compare the number of colonies which are growing on your on our plate to that acceptable limit, and depending on that we give pass to a product or fail to a product. Now if microbial count is below acceptable limit, then product is considered to be good and safe. But if the microbial count is above acceptable limit, it may indicate a problem with the product or the manufacturing process. So you have to fail that batch and you have to go for further investigation that why the microbial count is more. Okay, now the results of microbial test can also be used to identify the types of microorganisms which are present in the sample. Now if you find that uh, specific one or two type of cultures, they are more in number, which are growing on your plate, than using the pathogen testing or some other identification test, you can identify the type of microorganisms which is present in your sample, and then you can report and you can take action on that. Okay, so this can be accomplished through the use of selective culture media which are designed to support the growth of specific microbes. That is what I just explained. Okay, now after MLT, there is another test, that is bioburden test. Okay, so you also perform environmental monitoring. So bioburden test is actually a part of environmental monitoring. Okay, so but there is difference. So what is bioburden test? So bioburden, okay, that means biological burden. Okay, So it is a quality control process. That means QC member performs this process that detects and quantifies microbial contamination of a product at different stages of production. Okay, so biological burden is tested in your sample, raw material, then in process, and again in finished product, or say environmental monitoring is also done. So these tests can be used uh, both, you can say they can be run parallelly to see what kind of biological burden is there and whether the quality is getting hampered or not. Okay, so biological burden is very important test of the MLT. Why? Because it quantifies, detects and quantifies microbial contamination of a product at different stages of production. Now, after bioburden, there is environmental monitoring. So, what is EM? EM is environmental monitoring, and it is a process that determines the quality of controlled environment. Okay. Now, for example, you are working in a clean room area. Okay. Mostly the clean rooms are in QC, then in the production or packing area. You will find the clean rooms mainly used. So that is the control environment where the work is done. Okay, so microbial data collection is very important when you are working in a clean room, okay, or a controlled environment. So it is the process that determines the quality of that particular controlled environment. So what all or what is tested? So environmental monitoring, in environmental monitoring you will find air monitoring, alert and action levels to be noted down. Then surface monitoring is done. Non-viable contaminants are tested, viable contaminants are tested. The gowns or uh, sterile gowning is done or not, or what is the procedure followed, are the gowns sterilized before and after use, everything that is also monitored. Then clean room monitoring, then incubation, the area where the product is incubated, or in case of QC where the plates are incubated, what is the air quality inside that incubator. Okay, because it can happen that the air inside incubation incubator is actually contaminated, and that is giving false results. Okay, so that can also happen. So that is also a control environment which is to be tested. And again, in case of injectable products, uh, clean rooms are used where controlled environment is present. So again, environmental monitoring is to be done and followed. Now, what is difference between EM and bioburden testing? Now, bioburden testing, it is, as I said, actually a part of environmental monitoring, but it is the quantification of live microorganisms and identifying the microbial burden in the sample. Environmental monitoring is quantification of microbes to control the microbial acceptance criteria for manufacturing environment. So, in case of environmental monitoring, we are just um, looking, or we are just testing for the controlled environments. Okay, we are looking for the microbial load and its acceptable criteria. In case of bioburden, we are testing samples. Okay, that's the difference here. Now if you have done uh, bioburden testing or environmental monitoring, then you must have used Rodak plates. Okay, so this is a Rodak plate. Rodak plate, it has a elevated surface. Okay, it is not flat as our normal petri plate. These are small in diameter and the surface is slightly elevated. Okay, we will see about that. Before that, in case of settle plate method of environmental monitoring, the plates are exposed for how much time? So we expose plates for 4 hours. Okay, and after that we incubate them and interpret our results. Now, after this, let's move towards the RODAC plate. So, RODAC stands for Replicate Organism Detection and Counting. Okay, we replicate organisms which are present on a surface for their detection and counting them. That's the use of RODAC plate. So, they are used to take a sample from any surface to determine microbial load on that surface. If you are not using Rodak plate, then the alternative method for this is swab sampling. Okay, so you use sterile cotton swab, you dip it in sterile solution, and you squeeze out the excess saline solution, and you rub that particular swab on the surface by 5 by 5 centimeter square, and again you dispense that swab in the sterile saline solution. Okay, and you try to disperse whatever is collected by that particular cotton swab in your saline solution, and then you test that particular saline suspension. Okay, so alternative is swab sampling, and mainly nowadays Rodak plates are widely used everywhere, but if not available, you can go for swab sampling as well. Okay, so that's all for now. So these are all questions that I could think of. So it's not possible for me to upload each and every question because the video gets lengthy and you will get bored of. So I'm trying to upload videos in parts. So if you are struggling with any other question, do mention that under comment box. I will try to answer. And if you are struggling with any other topic, or if you want a video on any other concept, theory concept, practical concept, do comment that in comment box. I will try to upload a video. Okay, so I hope this video is helpful. Thank you for watching. Give a thumbs up, do like my videos, do share these videos with your friends, and do subscribe to my channel. And yes, don't forget to hit the bell icon so that you get notified on each new video that I upload.